In the last lecture, we went over the 4 types of organic molecules that you're made of: proteins, carbs, lipids, and nucleic acids. We just gave you a general overview of those 4 types of molecules. And then we got into the specifics of carbohydrates. And we ended with this page. Now we're going to get into the specifics of lipids. And so let's kind of compare and review a little bit the structures of carbs, and then we're going to talk about the general structure of lipids. So carbohydrates, we said that these molecules can either be classified as monosaccharides, disaccharides, or polysaccharides. So there's 3 types of carbohydrates to know. With lipids, there's 4 types of lipids to know. Now, with all organic molecules, they're made of monomers. And so the monomers of lipids are called fatty acid chains. Sometimes they're called hydrocarbon chains, and I just have a diagram of one here for you. And our 4 types of lipids are the triglycerides, phospholipids, the waxes, and the steroids. So we're going to start with the triglycerides, and as you can probably guess, we're dealing with 3 fatty acid chains. There's going to be 3 fatty acid chains. Phospholipids have 2, waxes have 1, and steroids have 0. Now, I'm more concerned, I guess, with the structure of triglycerides and phospholipids and steroids, not so much with being able to recognize the structure of a wax. Whenever you see a triglyceride, sometimes they're drawn with kind of a head region, and then you see 3 fatty acid chains, or sometimes they might be drawn circular, but you'll be able to recognize them because of those 3 fatty acid chains. With the phospholipid, you're going to see 2 fatty acid chains. Waxes, like they said, there's just 1 fatty acid chain hooked onto an alcohol, but again, I'm not too concerned about being able to recognize the structure of a wax. And steroids. Steroids have 0 fatty acid chains, and I'm going to butcher this drawing, but you'll recognize them because they'll have 4 fused carbon rings. Let me move the board up a little bit. But you'll see 4 fused carbon rings. So when we see that, like I said, I'm doing a very poor job of drawing this, but when you see something like this, 1, 2, 3, 4, then you know that you're probably looking at a steroid. Also, to help students memorize that steroids are the class of lipids that have 0 fatty acid chains, I tell students to think of, if someone has to take steroids to be really physically fit, that's like cheating in my book, and so you're a big 0 if you have to take steroids. But I also want to point out there's lots of other molecules that are considered steroids, and they're not all bad for you, maybe like anabolic steroids. And we'll talk more about steroids here in a little bit, but we're going to move on down to the pages that deal with lipids. So lipids is the more scientific term, but they also can be referenced as fats or oils. And as you all have learned, that fats and oils are usually considered non-polar molecules. However, there are some exceptions, and those exceptions include the phospholipids and cholesterol. Because both phospholipids and cholesterol have a polar region and a nonpolar region, so that polar region gives the ability to interact with water. We said that the elements that make up fats are carbon, hydrogen, and oxygen, and we said that you can recognize them different from carbohydrates if you look for that really high ratio of carbon to hydrogen. So there's a higher ratio of carbon to hydrogen compared to oxygen in a fat. And remember, those phospholipids, they have those phosphorus atoms. So they're kind of the exception. They also have phosphorus there. This concept that I've noted here at C is a concept that you're going to see in many units. And so when you have variation, you have then molecules that can provide many different functions. And you have the ability to be more flexible and respond to a changing environment. If you have lots of molecule variations, each molecule might be able to perform a different function for you. There's lots of different lipids that vary a little bit in their structure, but that variation provides you a molecule that has a different task or it has a different function and can provide a different function for your body. So we have some lipids that provide energy. When you change the structure a little bit, then you have lipids that provide you a different function, like the phospholipids. They provide membrane structure. You remove some of the fatty acid chains, and then you have lipids like the steroids, and many steroids are hormones. Hormones are signaling molecules in the body. Now, before we get into the triglycerides and the phospholipids, etc., let's take a closer look at these monomers, these fatty acid chains. Now, fatty acid chains can come in basically two main forms, saturated or unsaturated fatty acid chains. So let's take a look at the difference between these two chains that I have here. So first of all, if you take a look at the carbon atoms in this hydrocarbon chain, you can see that every single carbon atom is bonded with 4 other atoms. And so we say that it's saturated. This carbon atom cannot bond with any other atoms. The maximum that it can bond with is 4, and it has bonded with 4 others. This is what a saturated fatty acid chain looks like, and you can see there are no double bonds. So let's see what a double bond looks like. If we come over here to this chain, which is unsaturated, you see these double bonds. So here's a double bond, here's a double bond. If you think about it, this carbon atom, it has the ability to bond with another atom here, but it's only bound to 3 atoms, not 4. So we say that it's unsaturated. Now, you probably heard of polyunsaturated fats, and we're just dealing with fatty acid chains that have a large number of double bonds in them. So many double bonds equal a polyunsaturated fat. Poly means many. Now, we're going to reference saturated and unsaturated fatty acid chains as we go throughout these 4 types of lipids. So let's jump into the triglycerides. So when you think about using fat for energy, or if fat is being transported around the blood, then we're talking about a triglyceride. Triglycerides are the most common fat in the body. And they're primarily used for energy. And when you have too much energy, you've taken in too many calories, then those triglycerides can be stored in your fat tissue. So it's the form of fat that is stored in your adipose tissue, so in your fat cells. Fat has the ability to provide a ton of energy. As a matter of fact, it can provide twice as much energy as the same mass of carbohydrates. So it's great for storage. But it also provides a few other uses. First of all, it provides insulation, and that helps in temperature regulation. A layer of fat keeps you warmer, and some organisms that live in really cold environments have extra layers of fat to help them regulate their body temperatures. And it's also providing protection. It's actually providing a layer of protection around your internal organs. But there is too much of a good thing, and if you have excess triglycerides in your system, then lots of times you can suffer from coronary heart disease. And I'm going to show you some pictures in class of arteries that are clogged with fat. And this is what we mean by coronary heart disease. Now let's reference back to these saturated fatty acid chains and these unsaturated fatty acid chains. So I have 2 different pictures of triglycerides here. So here's a picture of some triglycerides, and here's some triglycerides. And you can notice that there's a difference here, that these have fatty acid chains that are straight, whereas these have fatty acid chains right here that are kinked. At that kink, there's a double bond. So I'm showing you these are triglycerides that are considered saturated fats because they're fatty acid chains. They're saturated. There's no double bonds. And these type of fats, these triglycerides, these saturated fats, these are solids at room temperature. Now let's talk about why they're solid at room temperature. So if you notice, there's no kinks in the fatty acid chains, And I can really pack these molecules close together. And in a solid, molecules are closer together than whenever they're in a liquid. The fact that there's saturated chains and there's no kinks, it allows these fat molecules to gather very close together to form a solid. These fats that are saturated are considered the bad fats. Like I said, you can tell a bad fat if it's solid at room temperature. So think about maybe some different fats that we fry food in. Butter is a solid at room temperature. It's not a good fat. Crisco, you have to scoop it out of the container. Those are saturated fats, and they're solid at room temperature. Now, your good fats are the fats that are unsaturated. And simply the reason why is because they're going to stay more spread apart, and they're not going to come together and form solids. So these unsaturated fats are going to be liquids at room temperature. So if you take a look at their structure again, you can see that with these kinks, they can't really get close together like these saturated fats can. So again, good fats are going to be the ones that are liquids at room temperature. Think of like olive oil and vegetable oil and canola oil. And so here I have a diagram so that you can understand these are good fats. If we're going to choose a fat to use, we want to try the good ones that aren't going to clog your arteries and try to avoid some of these bad ones. Now you have to be able to recognize structures of these different molecules. So when you're looking for triglycerides, then look for two things. Of course, you're going to see those 3 long fatty acid chains, but then there's another part, and you might hear the name of it, or they might label it for you. If you see that glycerol, that molecule that these fatty acids are hooked to, then you know that you're dealing with a fat. So there's a couple diagrams that show you structures of triglycerides. Next, we're going to get into the phospholipids. And remember we said that they have both a polar region and a non-polar region. Now, molecules that have both a polar and a non-polar region are called amphipathic molecules. And the polar head region is going to be hydrophilic. It's going to interact with water. Those tails, which are nonpolar, are hydrophobic. They will not interact with water. And because of the nature of this phospholipid, because of the polar and the nonpolar regions, those phospholipids will come together to form a bilayer. That membrane, that's a bilayer. Because the inside of the cell contains water, the outside of the cell contains water. We say that those are aqueous solutions. And so these polar regions are going to interact with water, and the nonpolar regions are going to interact with one another. So you get this phospholipid bilayer that makes up the cell membrane. Now, when you're looking for the structure of a phospholipid, they're usually pretty easy to identify. Again, you're going to have a glycerol molecule, so that's why I said if you see a picture and you see that word right there, you know that you're looking at some type of lipid. So, oops, I wrote that in the wrong spot. So there's that glycerol part of the molecule, and always look for that phosphorus atom, and you can see it right there. So that's the phosphate group. And then, of course, you're going to have 2 fatty acid chains. So I have 3 different diagrams here, and it's just showing you that there's a lot of different ways that they can diagram phospholipids. But again, here is that phosphate group, and then you have your 2 fatty acid chains. Here's the glycerol, and same down here. You have that phosphate group, glycerol, and 2 fatty acid chains. So moving on to the waxes. Waxes are important because they offer protection in most cases. Protection from pathogens getting into the organism like bacteria and fungus. That's what your earwax is doing. Bacteria or fungus that gets in there, it gets trapped on that wax. So it's offering you some protection. And it also gives some organisms a better ability to retain water. Your skin produces a wax, which is called sebum, plants produce a wax. It's called a cuticle. So if you've ever rubbed a plant leaf and it feels waxy, it's because there's a wax that's covering that leaf surface. You can see it right here on the diagram. This kind of acts as a water barrier, so it helps that plant keep water inside, and that water is not going to evaporate through the leaf like it would if it didn't have that waxy cuticle on it. Now, again, I'm not so concerned with recognizing the structure of a wax. I have a picture of a wax here so that you can see that there's one fatty acid chain. Again, just recognize that there's one fatty acid chain and that they're used primarily for protection. All right, moving on to the steroids. The steroids include molecules like cholesterol, and many hormones in the body are considered steroids. So that's why I mentioned that not all steroids are bad molecules. There's many molecules in the body that are classified as steroids that are necessary. So some steroid hormones maybe to recognize, like estrogen, progesterone, testosterone. Again, I would have a knowledge of at least one type of steroid hormone that you can discuss, because sometimes you're asked to identify a molecule and classify it and discuss the function of that molecule. First of all, let's jump into cholesterol here, because cholesterol is a major structural molecule of the cell membrane. So I have a picture of the cell membrane for you, so you can see it's primarily made of phospholipids, and this is that cholesterol molecule. And so you can probably tell already, just from what you learned in pre-AP, which part of that cholesterol molecule is polar and which part is nonpolar. So the nonpolar region is going to be interacting with those tails. So this is the nonpolar region. And then this region right here, which is going to be on the outside interacting with water, there's the polar region. Remember, this was the term that we discussed for molecules that have both a polar and a nonpolar region. The function of the cholesterol in that membrane, if you think of a fat, like think if you've ever made pot roast, and whenever the pot roast is warm, then you see that the fat is liquidy. But however, if you put that pot roast in a refrigerator, then you see that fat will start to form on top and it turns into a solid. So if you think about it, our cell membrane is made of phospholipids. It's made of fat. And the fluidness, or how solid the membrane is affected by temperature. So in cold temperatures, your cell membranes could become real solid. But in warmer temperatures, they could fall apart. They could become really liquidy. So these cholesterol molecules kind of act as a temperature buffer. In cold temperatures, when fats turn solid, that cholesterol actually helps the cell membrane become more fluid. But in warm temperatures, whenever fats normally turn to liquid, that cholesterol helps that cell membrane stay more of a solid. So it kind of helps act like as a temperature buffer. Now, this next concept you're going to see in several units. We have, like I've mentioned, that some of these steroid molecules are considered hormones. And there's two main types of hormones, I guess you could say. And I have two pictures here to show that. There's some hormones or signaling molecules that are water soluble. And oftentimes they're proteins, like insulin is an example of a protein hormone. And then we have some hormones, like these steroids. Here's an example of a steroid hormone. And what these hormones do is they're made by one part of the body, they're sent to the bloodstream then carries them to a different part of the body, and these signaling molecules can cause cells in those areas to respond. So let's take a look at kind of this first diagram right here. So we can see this protein-based hormone is moving through the body and it eventually ends up at a cell, at this target cell. And it's going to cause this cell to respond. And it does that by binding to a receptor protein that's on the surface of that cell membrane. But let's look over here and see what the lipid hormone does. So again, it's made in a different part of the body and it moves through the bloodstream and it arrives at the target cell. But you can see right here, it's not binding to a receptor protein. It's actually moving right through the membrane, and it's actually going to bind to a receptor that's on the inside of the cell. So those are two big differences on how these two different types of hormones can signal a cell to respond. So these steroid hormones can move right through that membrane because they are nonpolar molecules. Remember, the membrane allows, three types of molecules to cross easily. They have to be really small, they have to be uncharged, and if they're nonpolar, they can cross that membrane. So these nonpolar steroids, these hormones like testosterone and estrogen, they're going to cross the membrane, and then they'll bind to a receptor that's inside of it, and not just bind to a receptor that's on the surface. When it comes to the structures of steroids, look for, like I mentioned, those 4 fused carbon rings. I attempted to draw it on that first page for you, but there's going to be 0 fatty acid chains. So here's the 4 fused carbon rings that I mentioned. Now, since this is a short lecture, we're going to go ahead and cover the basics of nucleic acids. And I just want to point out, there'll be another unit in second semester. We'll review DNA and RNA and get more in depth. But this is just jogging your memory about what you already know and adding a few more details. Again, for all organic molecules, there are monomers that make up these organic molecules. And the monomers of nucleic acids are those nucleotides. Remember, we got out the little pieces of paper, and each piece of paper is called a nucleotide, and you would have to build DNA for me in pre-AP. When you look at the elements that make up these nucleotides, you're going to see CHOPN, C-H-O-P-N-N. Nucleotides and nucleic acids are made of carbon, hydrogen, oxygen. There's phosphorus atoms, and there's nitrogen atoms. And these nucleotides come in 3 parts. There's that phosphate group. There's the sugar. Sometimes it's called the 5-carbon sugar, and it's simply because there are 5 carbon molecules that make up this sugar. And then you have the nitrogenous bases. So there's a phosphate, there's a sugar, and there's a base in one nucleotide. And I have a couple diagrams of nucleotides for you. So again, we're seeing the phosphate, there's the sugar, there's a base. Here's the phosphate, here's the sugar, and here's the nitrogen base, and you can see why they denote it as the nitrogen base because it has nitrogen atoms in it. Now, there's lots of different types of nucleic acids. We talked about some of them in pre-AP, but not all of them. There's DNA, there's RNA, and there's different types of RNA. In pre-AP, we talked about messenger RNA and transfer RNA, important in transcription and translation of protein synthesis. Ribosomes is where you're going to find ribosomal RNA. And we're going to talk way more in depth about this called RNAi in our DNA unit. I just want to point out again, you have lots of variations of molecules, and these variations can give you a wider range of functions. So you're able to do more things in your cells because you have different molecules with different structures that all have different jobs. So let's just jump into DNA, and it's probably the most familiar to you. DNA stores your genetic information. It's found in the nucleus of your cells. And DNA is known to code for your proteins. And it does so by the order of these bases. And there was 4 nitrogenous bases, adenine, thymine, guanine, and cytosine. And you probably remember that A goes with T and G goes with C. And some students remember that the stick letters go together and the curvy letters go together. Now, some additional information for you. If you notice, let's come over here and let's look at adenine and let's look at guanine. And you can see they have similar structures. There's 2 rings to them. So here's a ring and here's a ring. They have a different structure than that thymine and cytosine. So here's thymine, here's cytosine. They're single rings. And so since adenine and guanine are both double ring, we have a name for them. They're considered purines. And cytosine and thymine are considered pyrimidines. Now, these are 2 terms that you're going to have to know. And I'm going to give you a way to remember that. And so kind of bear with me, because sometimes I have to come up with stories or have to come up with kind of off ways to remember some of these terms and what they mean and what things go with what terms. Where I grew up, it was a very rural community. I only had 38 students in my graduating class. And the ag school was really, really big. And so people that took a lot of ag classes, we called them Aggie. Think of students that, wear a lot of camouflage and always wear cowboy boots, those were the Aggies. And you could tell that a person was an Aggie, a pure Aggie, by looking at them, because they would have their cowboy boots on and they would have some camouflage. I remember that the purines are A and G. I think of those students that were pure ag students. So I remember purines, pure goes with A and G, those pure ag students. To remember that the purines are the ones with the double ring. Let's take this a little bit further. So lots of times ag students dated one another. Picture two ag students graduating and getting married, and they're die-hard ag students, so think of a lot of camouflage at the wedding, and they have a double ring ceremony where they exchange rings. Okay, so this is a really kind of a long story, but whenever you're trying to remember, you know, which bases are purines and which ones are pyrimidines, I want you to think of those pure ag students. So purines go with A and G, and I want you to picture two of them getting married. And so they're having a double ring ceremony. So think of adenine and guanine are the bases that are double ring. Cheesy, but it works. So our bases are found kind of more on the interior of our DNA, which you're familiar with. And we've learned that the bonds that hold these bases together are those hydrogen bonds. Now, if you look at the diagram, you're going to see a difference in the number of hydrogen bonds that form between these bases. Adenine and thymine are going to form 2 hydrogen bonds, whereas cytosine and guanine form 3 hydrogen bonds. So think about which bases do you think are easier to break apart and which ones are harder to break apart. So because cytosine and guanine, they form 3 hydrogen bonds between the bases, it's a little bit harder to denature DNA in an area where there's a lot of cytosine and guanine bases. Whereas if there's more thymine and adenine bases, which are only forming 2 hydrogen bonds, that's going to be weaker. And so the DNA is more likely to denature in those areas if there's a high concentration of adenine and thymine. And I have that written here. Sometimes it takes tricks to try to remember this information, because there's a lot to memorize in this class. To help me remember which bases have 3 and which one has 2 hydrogen bonds, I usually remember that cytosine, that's the 3rd letter of the alphabet. Cytosine is the one that's going to form 3 hydrogen bonds, and it does so every time with guanine. We already mentioned the sugar in DNA is called deoxyribose, and that's where that D in DNA comes from, deoxyribonucleic acid. You can tell from the diagram that DNA is double-stranded. Now, we say that the strands of DNA are complementary. They don't match each other, because if they match, then if there was an A on one side, there would be an A on the other side. They're complementary because the base on one side goes with the base on the other. So A is complementary to T, G is complementary to C. If I ask you what's complementary to peanut butter, you're going to say jelly. They go together. They're not the same things, but they're complementary to one another. Now, looking at that diagram of DNA at the top, if you look at one side and then look at the other side, it looks like this side over here is backwards, or it's upside down compared to the other side. And so these 2 strands of DNA are running in opposite directions. And the term for that is they're anti-parallel. If they were both running in the same direction, then we'd just say they're parallel strands. But since one is flipped and running in the opposite direction, we say that they're anti-parallel. And if you look at the ends here, so let's take a look at this end of the strand, and let's take a look at this end. And you're going to see that these ends look different. And because they look different, we denote them with different symbols so that we can reference, or we can talk about the different ends of a nucleotide. And so this end down here is known as the 3' end, whereas this end up here is known as the 5' end. And so since these strands are parallel, if this is the 3' end of this strand, when you look at this strand over here, you're going to see the 5' end, and then up here, you're going to have the 3' end. So these ends of these nucleotides are denoted with a 3' and 5'. So now we need to remember which ends are considered the 5' and which ones are considered the 3'. And so if you see that the end is ending and it's got a sugar, that's going to be the 3' end. And then if you can see up here, it looks like it's ending kind of with the phosphate. That's going to be the end that's denoted as the 5' end. And to help students remember that, there's one hump in a 5, there's one hump in a P. So you can take that 5 and you can kind of turn it into a P. I think that's what helped my pre-AP students remember that the 5' end is going to go on the end that looks like the nucleotide is ending with a phosphate. And that 3' end is going to end where it looks like it's ending with a sugar of that last nucleotide. We've always highlighted that the bases are held together by hydrogen bonds, but how are these nucleotides bonded together? So when one nucleotide hooks onto another nucleotide, or one monomer joins another monomer, we're going to have a condensation reaction, or dehydration synthesis occurs, and water is released, as you can see in this diagram right here. So to hook, here's one nucleotide. So if you want to label that a nucleotide. And here's the other nucleotide. And so for them to hook together, we're going to have to lose a water molecule. And you can see it being lost right here. Now this bond, there, kind of where that phosphate group is known as the phosphodiester bond. Now, I don't necessarily see this as a term that you're going to have to write about in a short answer question or on an essay, but if you have a problem and it's talking about phosphodiester bonds, then you know you're talking about some kind of nucleic acid there. And remember, all monomers, like when two nucleotides are hooked together, when a fatty acid chain is hooked onto a glycerol, when two monosaccharides are joined, we're making a bigger molecule. And so we make that through the process of dehydration synthesis. We talked about that in the previous lecture. And so I just want to show you that this dehydration synthesis happens in all of these organic molecules when we're trying to take monomers and join them together to form a bigger molecule. So let's talk about RNA and the differences between DNA and RNA. If you remember from pre-AP, we talked about you can recognize DNA because there's going to be thymine in it, but if you see U's, those U's are standing for uracil. So RNA does not contain thymine, but in its place it has uracil. Instead of deoxyribose as a sugar, it has ribose, and hence the R in RNA stands for ribonucleic acid. Instead of being double-stranded, RNA is single-stranded. So if you look at a diagram, you should be able to tell without even noticing the labels that this molecule is DNA because it's double-stranded, and this one's RNA because it's single-stranded. And again, there's many types of RNA, but that's good because it gives us lots of different variations and lots of different molecules to do different tasks for the cell. In pre-AP, we talked about the mRNA is important because it copies the DNA. So we're making the mRNA there. It's a copy of one side of the DNA strand, and then it's going to leave. And so here's our mRNA molecule. So the mRNA molecule is going to leave the nucleus, and then when it gets there, then we're going to involve these other molecules right here, and those are the tRNAs. Those tRNAs are going to bring the amino acids. So you can see that there's an amino acid right here that's hooked on to that tRNA, and it's going to bring it, and it's going to help these amino acids line up to make a protein or a polypeptide chain. So that mRNA is made in the nucleus. It's going to leave. It's eventually going to go out into the cytoplasm and find a ribosome. And then the ribosomes and the tRNAs and the mRNAs are going to interact to make this protein. And that's kind of just a crash course in what we learned in pre-AP. But we will have another unit again on protein synthesis, and we'll get way more in depth. So those tRNAs, again, they carry the amino acids to the ribosome where they're assembled into a protein. And I mentioned briefly that ribosomal RNA is a structural molecule because it's used to build ribosomes. So ribosomes are built of two things. There's that ribosomal RNA that's built into it, and then there's also some protein in there. RNAi, that I stands for interference. I just kind of showed you an overview of protein synthesis, and there's lots of ways we can stop this process, that we can stop the mRNA from leaving the nucleus. If the mRNA has left the nucleus, we can actually stop it from being translated. And that involves these other RNA molecules, which we call RNAi molecules. And there's two different types, and again, we're going to talk more about those in the protein synthesis unit. But basically, RNAi is involved in stopping protein production and silencing the gene. So when protein production is stopped, we say that the gene is silenced. So again, it has something to do with protein synthesis, but in this case, it's going to aid in the stopping of protein synthesis and silencing or turning off the gene, so no protein is being made according to that gene. And so now we're going to get into the third type of nucleic acid. So we already hit DNA, there's RNA, and now we're going to look at ATP. And ATP is used for energy in cells. When cells need energy, then they can break down ATP, and it releases some of that energy that that cell can use. Now, ATP is produced through that process of respiration. And you should have this equation memorized. So glucose and oxygen are utilized to make ATP molecules. In that process, CO2 is made as a byproduct, and so is water. Now, how this ATP is made in respiration it actually involves kind of a different form of the ATP, an ADP molecule. And really what happens is that mitochondria, where we say respiration takes place, it's able to take ADP and it's able to add this third phosphate onto it. So you can see that ADP has 2, and that's where the di stands, or di comes from, and ADP, di stands for 2. So ADP has 2 phosphates on it, and then we add that third phosphate onto it, and now we have a molecule of ATP. And that takes energy to do that. And the energy to take ADP and hook a phosphate onto it, that energy comes from your food. And so that's why we have glucose as one of the reactants in this equation. So let's take a closer look at the structure of ATP. It's actually a single nucleotide. It's not a macromolecule. It's not a big molecule like DNA. DNA can be like 4 billion nucleotides long. So it's a huge molecule. But ATP is just a single nucleotide. It's got a phosphate group, it has a sugar, and it has a base. But instead of just having one phosphate group, it actually has 3 of them. And so looking at this diagram right here, we've got, this is your phosphate groups, denoted with the P. Actually, I'm going to go up here to this part where it's showing you ATP hooked together. So here's your 3 phosphates, here is your sugar, and here's your base. And that base in the situation is adenine. We know that the mitochondria takes ADP and another phosphate and hooks them together to make ATP, but when the cell needs that ATP for energy, then the opposite happens. And we see that this third phosphate is removed, and as this process occurs, energy is released, and that energy can be utilized for the cell. Because we're breaking apart a molecule, remember that process is called hydrolysis. In this situation, we have to add water to break off that third phosphate. Again, then we'll be back to ADP, with this removed phosphate, and ADP stands for adenosine diphosphate. Again, that di means 2. ATP is utilized for energy in cells, but it's also involved in cell signaling. And cell signaling is a huge concept in AP biology. Students will tell me that on their AP exam, they feel like there was a lot of questions about cell signaling. We're going to learn a little bit about cell signaling in these first few units, but then we're actually going to have an entire unit dedicated to learning about the different aspects of cell signaling. But since we're talking about ATP here, let me kind of introduce cell signaling to you, and then you can see how ATP is involved in that process. So I'm going to start by referencing this diagram at the very bottom here, so you understand what this is showing you. So we actually have a cell here, and this is the cell membrane. And I mentioned earlier that there's receptor proteins that are embedded in this cell membrane. And hormones can bind to those receptor proteins, and the cell can respond. So you can see that the cell is going to respond here. Let's review a little bit, because we said that there's different types of hormones. There's the water soluble, and then there's the lipid soluble, or the nonpolar signaling molecules. Can you tell which type of signaling molecule you're seeing here? Well, since it's showing you that this signal molecule is binding to a receptor protein on the surface, that means a signaling molecule cannot enter the cell. It is not nonpolar. So you're dealing with probably a polar molecule or a protein hormone that is not going to be able to enter the cell. But whenever this protein hormone binds to this receptor protein, it kind of sends off a series of reactions that you see here. So I like to think of the game Mousetrap. If you ever played Mousetrap, you know, Mousetrap is set up to respond. And whenever you give it that first signal, then it causes the next thing to happen, which causes the next thing to happen. And this is what we mean by a signaling pathway. Let me show you where ATP can be involved in the signaling pathway. So whenever this molecule, this hormone, binds to this receptor protein, it sets off a series of reactions. So this molecule activates this molecule, which activates this molecule. And this cyclase molecule can then, once activated, can grab onto ATP, can bind to it in its active site, and can actually convert it to a molecule which is called cyclic AMP. And cyclic AMP is known as the secondary messenger, and it's a common secondary messenger, and there's other types of secondary messengers. But this hormone was considered the first messenger, and the cyclic AMP is called a secondary messenger because it's going to actually signal a bunch of different molecules inside of the cell, in the cytoplasm. So cyclic AMP is known as a secondary messenger. And it's important in many signaling pathways that we're going to take a look at. Now, so let's take a look at how ATP is made into cyclic AMP. It's actually pretty simple. You can probably diagram already how ATP can be broken down into ADP. You just have to remove the phosphate. Well, if you remove 2 phosphates from ATP, then it's going to be left with 1. And that molecule is called cyclic. I'm going to go ahead and write cyclic AMP. And for short, we just put a C there and abbreviate it, cyclic AMP. So here is our ATP molecule with 1, 2, 3 phosphates. And to make cyclic AMP, then there's going to be a hydrolysis reaction. Water is going to be added, and those are the 2 phosphates are going to be removed. You can see them over here. And then we're left with this part, which is called cyclic AMP. And there's just 1 phosphate left on that molecule. We need to go back real quick before I forget to that page where we have the different structures. So again, we need carbon, hydrogen, oxygen, phosphorus, and nitrogen to make those nucleotides. And those nucleotides are the monomers. You should be able to recognize the 3 parts. There's that phosphate group. We have the sugar. And we have a base. There's 3 main types of nucleic acids. So we have the DNA, we have the RNA, and we have the ATP. You should be able to recognize the structures of both. Again, this one's easy to recognize. This is DNA. It's double-stranded. RNA is single-stranded. Know the difference in the sugars. DNA has deoxyribose, RNA has ribose. Know the difference in bases. DNA has A's and T's and G's and C's, whereas RNA has A's and U's and G's and C's. And then finally, your ATP is more like a single nucleotide. Instead of 1 phosphate, though, it's got 3 total.